Hello. We have to get to today with us Tim Dempster, Luke Barrett, and Andrew Coates, and all of them co-authored 2 papers for the special issue of International Journal of Parasitology. And we will discuss some of the content of the papers, but maybe start from something a bit more general. So Tim, could I start with you? Are you a fish parasitologist, or how would you describe yourself and how your interest in fish parasites started?
Well, I think it's fair to say I'm not a fish parasitologist. I know very little about parasites compared to most parasitologists, so I'm definitely feel like an interloper in this area. But I guess now for the past maybe 7 or 8 years, we've been working pretty intensively on salmon lice, sea lice in general, amoebic gill disease, and other parasites. And the way our lab approaches it is to try and come up with new ways of stopping them from infecting the fish in the first place, or better ways to treat them. So in that sense, we don't need to know much about what the harm they cause or how to diagnose them. We just need to know how to stop them or get rid of them. And so we're using sort of more ecological knowledge and knowledge about fish welfare and fish biology. So I'm more a fish biologist, a marine biologist. So yeah, that's our general approach to the systems that we work in. Yeah.
And in your opinion, which parasites are the most important for fish and communities who rely on fishing and aquaculture, and why are those parasites most important?
Well, look, I guess, communities that rely on aquaculture globally, we mostly work with salmon, and so there is really only one that most people talk about in salmon. There are, of course, many, but sea lice, whether they be salmon lice or caligus, are really important for salmon production. More globally, I'm sure that their ectoparasites are pretty important as well. So I guess that's our focus. Probably also they're more visible to, then they flow out from farms to wild fish, and that has a negative effect, and that constrains industry growth for aquaculture and also has an impact on wild fish. So they're really parasites that are at that juxtaposition between fishing and aquaculture, and the way communities perceive aquaculture makes them really important.
So just to clarify, you said sea lice were really important, and obviously globally they are, but do we have a problem with sea lice in Australian aquaculture?
Not much, no. So not in salmon. They're not absent. There are some things that you would call sea lice, but they've never caused much of a problem. They're very peripheral, and it's other pathogens. Pilchard orthomyxovirus now, plus amoebic gill disease, that are the main problems in that production. And they obviously they come from outside into farms, and they come from outside, but there's not a lot of evidence that amoebic gill disease is really problematic for wild populations. It's probably there. I don't think we know all that much about what it does in wild populations. You're the expert there, Barbara, so you could fill us in. And Pilchard orthomyxovirus, of course, was a terrible virus in terms of what it did to pilchard stocks across southern Australia many decades ago, and it's kind of disappeared and has obviously been there all along, and now it's been transferred from wild fish over into salmon production and is causing major problems. Yeah.
So going back to sea lice, and most of your research has been done in Norway when you work on sea lice. Traditionally, when sea lice issues started, they were treated with chemicals. Why did the chemical treatment stop? Like, was it just because people wanted clean and green fish, or are there also other issues with using chemicals to treat fish against parasites?
The simple answer is evolution. So it's the evolution of resistance to those chemicals. And so most of the major chemicals in Norway, in fact, probably all, and Andy can speak more to this, there's evidence strong evidence for some, and the actual genes that have appeared that, or of mutations that have appeared that have been expanded in the louse population that confer now quite strong resistance to some of these chemicals. Interestingly, it's not universal, that evolution of resistance. So if you go to Canada, they still only use one chemical and it still has very good effect. And that's because the evolutionary drivers in Canada are completely different to they are in the Atlantic. So, and that's got to do with the fact that there's a very big wild refugia of lice in Canada. So that buffers rapid evolution in farms, whereas in Norway you have lots and lots of farm salmon and lots of lice on farm salmon, and only a very small population of wild salmon, and so there's no buffering from the wild lice population. So they're all put under this pressure and get rapid evolution, and you can probably speak a bit more to that. So.
And obviously fish farmers have been looking for all sorts of different options when they realized they couldn't use chemicals forever. And you have this beautiful picture behind you of cleaner fish. I was just going to ask Luke, who is the first author of the paper on effects of cleaner fish on sea lice. Could you tell us more about your research on cleaner fish on sea lice and the implication for Norwegian salmon aquaculture?
Yeah, so cleaner fish is quite a cool concept, really. It's possibly the only example of a vertebrate being used to control parasites in an industrial context. And it was really picked up quite eagerly by the industry because they were trying to shift away from these chemicals, which were losing efficacy. There were also concerns around contamination of food and the surrounding environment. And cleaner fish were quite a neat solution to that problem. There's quite a good evidence base from smaller systems in, say, experimental scale cages, where within a couple of days to weeks, they can clean all the lice off, 500 or 1,000 salmon. What we don't know as well is whether they have this same effect at industrial scale. So it's quite a different system when you move from a 100 cubic meter cage up to a 10,000 cubic meter cage. So that was where our study came in.
And there are obviously more than one species of cleaner fish. So were you looking at both species or only one particular species of cleaner fish?
Yeah, so there are probably 5 main species. There's the lumpfish, which is what Tim has in his background. They're from the northern Atlantic, quite a cold water species. And we've also got a few species of wrasse, which are native to most of Norway, but they do a lot better in the southern half and also down into lower in Europe. And so in this particular paper, we looked at lumpfish versus wrasse. So we tried to see whether there was a difference in the detectable effect of those two types of cleaner fish. It looks like wrasse do a little bit better on average, but it's also hard to say because lumpfish tend to get a bit of a rough situation where they get put in the cages at a time when the wrasse won't survive. And so it's not necessarily a fair comparison in that respect.
And originally the cleaner fish were coming from the wild, but currently there are whole farms or hatcheries which breed cleaner fish. There are vaccines for cleaner fish. So there's almost like a production in parallel to salmon. They are also producing cleaner fish. So in your experiments, in your research, were you looking at farmed cleaner fish or wild cleaner fish?
At the moment, most or nearly all lumpfish are farmed. They've really managed to scale up that hatchery operation. At the moment, most wrasse are still wild caught. And so in this analysis, we didn't distinguish between farmed and wild caught wrasse. There have been some studies previously that suggest there's some differences, but it also varies a lot depending on the exact history or acclimation of the fish being used. So in this case, we took a very kind of industry-wide, kind of broad-scale view of the situation.
Mm. And what do you think is the future for cleaner fish and on salmon farms?
I think they have their place. I think their future is we need to get a lot better at using them efficiently. Our view from looking through the national database of cleaner fish use and lice numbers is that they're not always used at the best time of year. They're possibly, possibly, we don't know for sure, but we think maybe there's still some scope to improve the way they're used. So making sure that the fish that go in the cages are ready to go in the cages, they're at the right size, they've got things like hides and things in the cages basically to make their life better, help them live longer, clean more lice. And then if we can make individual fish more efficient, we don't need as many to get the same effect, which is, I think, a key goal.
Yeah, really interesting. So Tim, you said that the chemicals stopped being used against sea lice because sea lice could develop a resistance. Can they evolve in any way to make the cleaner fish less effective?
Well, this is a good segue for Andy's paper because, yeah, there is potential, of course. So cleaner fish are visual predators. They have to see the prey and then move in and pick it off the moving salmon. And so the less they're able, the less they're able to pick up that there's a louse there, the lower their feeding will be. And so you can think then, if you look at those sea lice behind Andy's head there, they have pigmentation spots, and there seems to be a natural variation in pigmentation. We don't know whether that has a genetic basis yet, but there have been reports that lice are getting lighter. So if you're a lighter louse, then maybe you're more translucent, you're harder to pick up by a cleaner fish. And if the selection pressure from the cleaner fish is pretty heavy, and it's over 50% of farms are using cleaner fish, there's more than 50 million stocked per year. So it's a fair selection pressure. So it's possible you could have evolution in that direction towards lighter colored parasites that are harder for the cleaner fish to see. And maybe Andy, you can talk about the possible way that could go.
Yeah, I guess I sort of hit the nail on the head there. And it's mostly, I guess, anecdotal now, all these stories of these light or these transparent lice emerging, but we are sort of starting to do a little bit of work on that. And there's, I think, a paper in the works from the team at the Institute of Marine Research in Norway that are finding that a potential plastic effect of the pigmentation in the lice, but that also could have like a genetic underpinning as well. And so exactly like Tim was saying, if these pale transparent lice have a better chance of survival against the cleaner fish, and then they can pass it on to their offspring, then in a few generations there is that potential for these more resistant lice to emerge on the scene.
And they can have 8 to 10 generations per year, so it can go fairly quickly. In a decade you can have 80 or 100 generations, and rapid evolution can certainly occur within that time.
So Andy, we've been just talking about the cleaner fish maybe not being useful for everything, and maybe the sea lice changing so that it's not as easy for the cleaner fish to find it. What are other non-chemical pest controls, and why are they important for parasite control in aquaculture?
Well, there's thermal delousing, which is bathing the salmon in warm water or hot water, which causes the lice to fall off. There's mechanical delousing, which is, I sort of picture it as like a drive-through car wash for salmon, where you have jets of water or little brushes that knock the salmon off, knock the lice off the fish. The one that I've been focusing on, which my recent paper is on, is focusing on lice skirts and snorkel cages. So these are actual modifications to the salmon cage itself, which are designed to prevent the lice from getting in the first place. And I guess that sort of age-old adage of prevention is better than the cure is exactly true for the salmon lice. And Tim and Luke have actually, I think, just got a paper coming out very soon on this topic. And so, yeah, these prevention cages or these preventative strategies are sort of the ideal, I guess, when it comes to lice management. But my paper is sort of talking about how even these strategies are not necessarily durable to resistance in the long term. Yeah.
So can you tell us more about salmon louse copepodids and in particular what's important about this parasite swimming behavior?
Yes. So I would love to. So the lice you see above me are adult female lice, and they spend most of their time attached to the salmon. They spend most of their time around their host, and the transmissive phase is their larvae. So they release hundreds of offspring to the water, and these larvae, which are called copepodids, drift in the plankton. And they can travel tens of kilometers at a time until they eventually encounter a new host, and then they attach and complete their life cycle as a parasite. And most of these copepodids spend their time in the shallow water, in the top several meters of the water column. And so those prevention cages I was talking about, they extend, they're basically walls that wrap around the cage for the top several meters and block all these incoming copepodids from getting into the cage. But at the moment anyway, cages still need water circulation for the welfare and the growth of the salmon. So the lower parts of the cage are still open to the water, and sometimes copepodids are deep enough in the water to actually slip underneath and get inside the cage. So my research was looking at whether the swimming behavior of the copepodid and their preference for depth is just a random feature, or if there is actually some sort of trait or phenotype related to this behavior that means some lice are deeper in the water than others. So that's, yeah.
And are they?
I mean, it looks that way, yeah. So my study was just looking at different individuals' families of lice that were reared in the laboratory. And I looked at their swimming behavior and their sort of preferred depth in the lab. And I did find that there was very high variation in these vertical distributions of the copepodids, and that some families preferred much deeper depths than others. And we can't rule out environmental effects, or we can't rule out maternal effects to this, but this does suggest that there might be a genetic component to swimming behavior, which means that these deep lice are getting into the cages and they're producing deep offspring. And you can get that picture emerging of these deeper swimming lice appearing in the population that are more resistant to these depth-based preventions.
So again, they may evolve and not be really sensitive to this type of treatment, and we may have to come up with a new type of treatment.
Maybe.
So what do you think is the future for? So would you suggest that the scares have to be longer, or that we have to come up with a different management strategies, or maybe combination of different management strategies?
I mean, I guess we can keep throwing new technologies as often as we can, but that's expensive and it's wasteful. So I think the key is really, as you said, to manage these technologies with a sort of evolutionary understanding in mind. So if we figure out where this selection is strongest, or find out which different combinations of strategies work synergistically, we can really kind of harness that knowledge and use evolution to our advantage, which I think that's definitely the future when it comes to lice management.
So how are you following it up with your research, this particular aspect?
A little bit, yeah. So I'm looking at the moment on, so what I was talking about before was the variation in the lice population in swimming behavior and how that might have a heritable component. And the other side of the coin is this trait actually selected for, which is what I'm working on at the moment. And like I said, if selection is really strong in some areas and weaker in others, then we can, put all our focus into using skirts and snorkels on those areas where selection is weakest. And on those areas where it's very strong, we might want to use a different strategy like cleaner fish. We might want to up the use of thermal mechanical treatments instead.
So in general, for pest control, not specifically in aquaculture, but in agriculture, even when chemicals were used, the strategy was to expose each generation of pests to different chemicals, so they couldn't potentially evolve resistance to chemicals.
Exactly,
because So would it be also something to consider that maybe, I'm not talking about chemicals here, but maybe change the management strategy so the generations are not exposed one generation after another to exactly the same management strategy?
Absolutely, yeah. And I think that was definitely floated a little bit when it came to the chemical pesticides that we used. But one of the big problems was that farmers just often didn't have access to multiple treatments at once. But hopefully we've learned our lesson a little bit and taken lessons from terrestrial agriculture. So if we can combine those different strategies on the one farm or the one area, and really the kind of multi-treatment use in one generation, that should, theoretically, I suppose, sort of mitigate the effects of resistance arising.
Yeah.
And there's and he's also identified that there are some techniques that push traits all in the same direction, and that's when they're synergistic, and that's a problem because then you get rapid evolution in that direction. But there are some techniques that are pushing in different directions on the same trait. And so the trait's not going to move, away from where you want it to be. So it's smart thinking about the combination of methods that are applying antagonistic evolutionary pressure on the parasite, so that they both maintain their effect. And Andy's done some really good thinking in that space.
So can you give an example, Andy, of what you would propose?
So I think the big takeaway from this issue at first is that there hasn't been enough research yet. So we definitely need more research into this topic, which is starting to emerge at the moment, which is great because, as I said, we really need this kind of evolutionary thinking to focus on the long-term gains of pest management in aquaculture and in farming in general, as opposed to the kind of short-term focus that has been occurring in the past. I mean, a great sort of hypothetical situation, I suppose, would be, we talked about the cleaner fish being lighter colored in response to the cleaner fish, the lice being paler in response to the cleaner fish. We don't know the reason why lice have those pigment spots, but it's very likely it could be for UV protection, which we see in all sorts of other crustaceans. So if we can somehow harness that and say, okay, these pale lice are better against cleaner fish, but they're more susceptible to ultraviolet radiation. Can we, prioritize the use during summer or during lower latitudes, or even, I suppose, install some sort of artificial UV lighting at certain situations that, as Tim said, can provide that stabilizing selection. It's the sort of the fitness costs to the resistance. And if those costs are stronger, the advantages of being resistant, that we can kind of keep the population in check.
So that's a really interesting approach, and you've talked quite a bit about using the skirts on the cages. I was just wondering if either you or Tim could talk about snorkels, so to explain what the snorkel cages are and how they are used against parasites.
Well, I can talk about them. So they're very much the same principle as a skirt. So the skirt works because it's an impenetrable skirt that goes around the top of the cage, and they're usually 6 or 10 meters deep. And then the water basically at the surface, where there's more lice than deeper, it just flows around. And so the fish, when they're up at the surface, they don't get infected. Now, the problem with skirts around an entire cage, and that might be a cage that's 160 meters in circumference, is that you reduce water flow. And so then you have the issue of lower oxygen, because the big biomass of salmon use up the oxygen. So skirts, particularly in summer and autumn, can be difficult to use because of poor oxygen, and it's bad for the fish. The snorkel is just it's essentially, you have to open the cage and the normal netting down the bottom, and then you have a roof that comes in with a little snorkel at the top. And the reason salmon need to get to the surface is they have an open swim bladder, and they only fill up their swim bladder by swallowing air at the surface. So they have this behavioral need to get to the surface, and they fill up their swim bladder, and then that keeps them neutrally buoyant in the water. And if they don't fill up their swim bladder, then they're always sinking. and they have to swim faster to not sink. They hydroplaning with their fins, and they burn too much energy. So, I say to my lab, you can't farm salmon underwater completely. They need the surface. So, yeah, so that's what a snorkel is, and you've only got then a small volume that's then shielded because the salmon are held deeper most of the time, and in that volume you might have poor water quality, but it's a much smaller volume, so you can then pump water in from the deep, or you can oxygenate, so you can take care of the water quality in that small volume, whereas you can't take care of it in a bigger cage. So that's the snorkel technology, and it's now, sort of the first one was an invention at 5 by 5 meter cage 10 years ago, and it's been commercialized by Agerson Net, which has been bought by Aqua Group, and now selling them commercially, and they're appearing on lots of different farms in Norway. So I think Moe just bought 7 farms worth of snorkel. They bought.
Wow, that's very impressive.
50 of them. So it's now a major commercial product. So it has much less market penetration than skirts, but it's emerging, and it seems to give fairly good and consistent results, the snorkel, and the results from the skirts can be very good, but also it can get a bit mixed as well, because the other advantage about the snorkels is that the water is flowing around. As the water flows underneath, it mixes a little bit into the bottom of the snorkel, and that's normal. When again you have a big cage and a skirt around it, and the water flows underneath, it mixes much more up into the skirt, so you can get parasites coming up into the skirt much more than you can into a snorkel. So all that sort of hydrodynamic testing has been done by others in Jeff that shows that's the case.
And would moving farming offshore help with the parasite management, or do you think it will create other issues? Because obviously when you're in an exposed area, it'd be much harder to put snorkels, skirts, or any other manual manipulations of the cages or fish.
I think, Luke, you want to answer this one?
Yeah, sure. So there are certainly fewer lice offshore, mostly because there are fewer salmon and salmonids producing them. But offshore farming has a lot of challenges as well, and we certainly haven't seen it take off yet. It's clearly going to be more difficult for them to compete in a marketplace where inshore farming is allowed. Things like the risk of working on these cages in big swell, the increased engineering effort required to make them stable in big swell, the fuel costs, just everything is harder. So it's certainly something that will be looked at and is being looked at. Remains to be seen whether it's a viable solution or not.
I mean, we can sort of preface here and say that we've just done analysis of how offshore salmon farming is globally, at all the countries in the world. And I don't know what the grand mean is, but the average salmon farm is about 250 meters from a coastline. Wow,
that's much closer than I expected.
Offshore salmon farming is fiction. So of course there are salmon farms in more exposed locations that have more waves and more current, but almost every salmon farm in the ocean in existence is tucked behind or close to something, and for logistical reasons that makes sense. It's easy to get, it's easy to work on, and also it's shielded from the forces of the ocean. So it's still the idea of open ocean farming is still quite futuristic in our view.
So potentially fewer parasites, but definitely more other challenges, and maybe the fewer parasites is not, maybe the cost of having the fewer parasites could be more than it's worth it, potentially.
Well, I would just simply say, if there was a really clear economic argument for offshore farming en masse, it would already be It would already be being done. But there are many that are trying to, build bigger farms that can operate in really exposed locations, and they can. But even those farms are usually not very far from land, because the simple fact is that you still need to get people to them. You still need to operate them, and putting something a long way out makes enormous logistical and occupational health and safety difficulties that as yet there aren't solutions for.
So what's next in Norwegian aquaculture research or changes in how to manage sea lice then? If the movement offshore is not really a viable option at this stage, what do you think would be a viable option, and where would you concentrate, or where are you going to concentrate your research effort?
I'll start, guys, because I can talk about a new project that we're involved in, which is being led by Nick Robinson from NOFEMA, funded by the industry, so the Norwegian Seafood Research Fund, and they're looking at things that might be a real leap, like a moonshot step in how to deal with lice, because if you look at across all the things being done at the moment, maybe that will kind of hold things where they are, but you can't project that people are really going to get a full handle on lice with the current suite of tools. They're looking at new tools. So the tool that this project is looking at is trying to find out why other species of salmonids don't get infected with salmon lice very much. Something to do with the morphology, physiology of their skin and their mucus layer, or other aspects of their biology. Trying to identify those mechanisms that mean that perhaps the salmon lice don't attach, or they don't sense the fish, or they don't like attaching to that fish. Working out the genes that encode those mechanisms, and then using CRISPR in Atlantic salmon to see if you can clip out those bits of the genome that make an Atlantic salmon attractive to a salmon louse. And so then you may have a situation where you've got a gene edited salmon that a salmon louse no longer recognizes as a host. And so
that sounds fascinating.
Yeah, so that's a sort of moonshot technology, and there's a major project that the Seafood Research Fund is invested in to try and do, the first steps towards that kind of approach. So that's a long shot technology, but one that could provide a great leap ahead.
Yeah, that's really amazing, using gene editing for applied aquaculture. Maybe Luke, what is the future of your research? Like, what are you doing next, or what are you working on now?
Yeah, so I'm interested in a few different aspects. It's definitely still continuing with the cleaner fish topic. Basically, I'd really like to figure out how we can get the most out of these cleaner fish, and how to do it in a way that safeguards their welfare as well. They're these are intelligent fish. They're probably smarter than salmon. Their welfare should be as high a priority as it is for salmon, and that's currently probably not the way they're treated.
There's so many videos on YouTube about training lamfish, and lamfish going through little rings underwater and doing all sorts of clever tricks. So they definitely must have impressed lots of people with their intelligence.
Yeah. Also, some species of wrasse have been seen using tools. They're they're clearly among the smartest fish in the ocean. So if that correlates with their ability to suffer, that's clearly an important point to consider when using them in industry, especially if there's alternatives. So yeah, in my view, I think we probably need them for now. We need to use them more effectively, more efficiently, use them in concert with other methods, so that together we have a suite of tools that really work well together, as Andy was saying, minimizing directional selection. And then we hope for the silver bullet in the future, or at least a significant step forward in terms of genetic resistance and so on.
Yeah. And Andy, you already talked about your future interest, but I was just wondering, and this is a really question for you first, but also Luke and Tim, how do you continue your research in Norway while we can't internationally travel? What are how do you deal with these challenges? Like, it's hard for everybody to, fortunately the Melbourne lockdown finished, but it doesn't mean you are going to Norway. Andy, how are you going to continue your research on sea lice when we don't have them in Tasmania?
Exactly, it's a big problem. So far, managed to cope though. Just been a lot of computer-based work, which I'm very fortunate enough to have tucked away all these questions and ideas that I can either just write about. I've been working on a lit review, but also computer modeling. There are some really incredibly powerful computer models that are used in Norway, especially, that track how the lice larvae, those copepodids I was talking about, are dispersed through the environment on ocean currents. And so I can actually sort of, by corresponding with the people in Norway, play around with that model and see how, if we tweak certain aspects of louse behavior, such as the swimming behavior I was talking about, how does that actually affect their dispersal through the environment, but also how does that actually affect their dispersal up and down the water column. So thankfully I've been able to test some of my hypotheses on my computer using these modeling techniques, which is very lucky. But of course I would like to get back into the lab as soon as possible and get back to Norway. Hopefully soon. We'll see how we go.
And what about you, Luke? Like, how can you work in Melbourne on cleaner fish?
Yeah, I mean, like Andy says, a big shift towards doing lit reviews. If the world,
so lots of publications.
Yeah. Hopefully the world still needs more reviews after this. And also been starting to spend more time with the kind of data science type parts of the work. So Norway has this amazing database of lice counts from the last 10 years almost.
Yeah, so we're blessed that there are amazing public data sets related to this work, and we've just been accessing those. We haven't done it before, but we should have, because what they can tell you is incredible, and at industry scale. So, and Luke's doing himself a disservice, but I think both Luke and Andy, strangely, have accelerated in their productivity under COVID, because they've had to sit down and analyse and write. We're hoping to get back there soon, but thus far we've been okay. And we've also, probably like most researchers, you sometimes have things that you haven't finished that are sitting on your desk or in your cupboard, and you've been wanting to get to it for a long time. So we've been digging into those data sets that we've constructed from a long time ago or a while ago, and working on those. So I think we'll be busy for a while, but we'd love to go back. But I think I don't think it will be next year, Barbara. I think it might be 2022 before we see the other side of the world again.
Well, I still hope we all will meet over a beer in Cairns next year, and with hopefully it'll be International Symposium of Fish Parasites, but even if it's Australian Society for Parasitology Conference, I hope we all be there. And once again, congratulations on your fantastic papers published in the special issue of IJP on fish parasitology. And thank you very much. Thank you to Tim Dempster, Andy Coates, and Luke Barrett from University of Melbourne for joining us today. Thank you.
Thanks, Barbara.
Thank you.